Hello students. The topic cell, unit of life, is coming to an end. At the end of this topic, let us study ultrastructure of nucleus. When the cell is not dividing, division is either of mitosis or by meiosis. So when the cell is not dividing, the cell is preparing for the division. At that time, the resting stage of the nucleus is known as interphasic stage. So at interphase, the nucleus shows following parts. It is usually spherical in shape, bounded by discontinuous double-layered membrane known as nuclear membrane. Now, due to discontinuity, we can see the minute openings in the nucleus. Okay, these are referred as nuclear pores. These are guarded by annulus. These work like gates. If you look at the nuclear membrane carefully, there are two layers, outer layer and inner layer. In between these two layers, we can see the perinuclear space. This will regulate the transport of components within the nucleus or the molecules which are moving outside the nucleus. Inner to nuclear membrane, the space filled with nucleoplasm. So here, instead of, this is wrong spelling, it is called as nucleoplasm. In the nucleoplasm, we can see one more spherical body. It is known as nucleosome. And there is a network of thread-like structures. This we call chromatin network. At the time of mitosis and meiosis, this chromatin network loosens and each chromatin fiber, which becomes thick and short, known as chromosome. Thus, in this topic, we are initiated with the prokaryotic cell, taking example of bacteria. Under eukaryotic cell, we have considered plant cell and animal cell with differences, and then we have learned cell wall, which is fully permeable protective layer absent in animal cell, present in rest all cells. Cell membrane or plasma membrane, it is semi-permeable in nature, and then inner to plasma membrane, we can see the cytoplasm with the nucleus. In eukaryotic cytoplasm, we can see the endomembranous system comprises endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. These vacuoles are of different types in the unicellular organisms like food vacuoles and contractile vacuoles. But in animal cell, the vacuoles, even if they are present, they are very small and inactive. But in plant cell, vacuoles are very active. There is a structure of mitochondria learned here, which is known as powerhouse of the cell. Then we have learned plastids under the heading chloroplast, leucoplast and chromoplast. Detailed structure of chloroplast is studied, and in today's session we have learned nucleus ultrastructure. In previous session I told you about the microtubules, microfilaments, interdigitated protein fibers which representing cytoskeleton. The extensions from the cell membrane are cilia and flagella. Cilia are hairy and they are numerous. Flagella are whip-like, they are helping in locomotion or trapping the components. This structure, what we have learned today, it carries 5 marks weightage. So these are the labelings compulsory to get the 3 marks and explaining these components which carry 2 marks. With this, I can say the detailed account of cell is completed. At the end of this topic, we have to learn the differentiated chromatin fiber, and that differentiated chromatin fiber we call it as chromosome, which are visible at the time of mitosis and meiosis. In mitosis and meiosis, there is one stage called metaphase. So in metaphase, if you see the structure of chromosome, it shows two arms. These are held together, okay, by a narrow region surrounded by commissural cap giving the fibrillar microtubules. This region of the chromosome is known as centromere. The two arms of the chromosomes, okay, these are known as chromatids. The tips of the chromatid we call telomere. There are some chromosomes in the given set can show the chromatids with the bulged telomere. Okay, this region is not found in all chromosomes. It is known as satellite. Chromosome with satellite is known as SAT chromosome. This structure is important for again 5 marks. A typical chromosome which is visible in metaphase. So this we call metaphasic chromosome. A metaphasic chromosome shows two identical arms known as chromatids. Both are held together by a narrow region known as centromere. Now this centromere is highly protected, giving primary constriction. If satellite is there, below that the narrow region, if it is found, then it is known as secondary constriction. So when you explain the structure of chromosome, remember a diagram showing two arms. These are bundles of the threads known as chromatids. The tips which are non-sticky called as telomeres. Primary constriction showing centromere and having commissural cap with kinetochores. Now when you magnify this chromatid, okay, we can see these beaded strings. Now when you magnify this beaded string, the beads are composed of a protein, name is histone, and total 8 histones forming an octamer. And around this octamer we can see this which I am showing in orange color. This we call DNA. This is again DNA which is wrapped again around the octamers made up of histones. Around octamer again the DNA encircles. Okay, and it comes out. Now this region octamer with DNA, this we call heterochromatin. Next year you are learning about this. This is heterochromatin, and this region where we can see only DNA, it is known as linker DNA. So heterochromatin composed of DNA and histone and linker DNA where histones are not found, this we call euchromatin. So basically the chromosome is composed of DNA packed with histones. Okay, DNA, histones together form chromatin. Chromatin are supercoiled forming chromosome, and chromosomes are known as vehicles of heredity. Okay, in the next session, we will classify chromosome. Again, in that video, I'll explain the detailed account of the chromosome. So from today's session, better you should be perfect in the structure of nucleus. Thank you very much.